Okay, so it's 12 o'clock, so then we'll get started. It's my distinct pleasure to introduce Lydia Finley today. Lydia did her undergraduate studies at Yale and then came to our very own BBS program where she worked with Marcia Haggis. She then moved to Memorial Sloan Kettering to do a postdoc with Craig Thompson, where she stayed on as faculty. She's now assistant member of the cell biology program at Memorial, as well as assistant professor at Weill Cornell Medicine. She's received really numerous awards. She's a fellow of the Damon Runyon Cancer Research Foundation. She's a Searle Scholar. She's been awarded the Pershing Square Sloan Prize for Young Investigators in Cancer Research, among many others. Lydia has had a long-standing interest in understanding how cellular metabolism regulates normal and malignant development. And again, it's really a pleasure to have you here, Lydia. We look forward to your talk.
Thank you so much for hosting me. I'm really excited to be here and have a chance to share some of the work we've been doing in the lab and hopefully have a nice discussion with people who've tuned in. Thank you for tuning in. So as you just heard, my lab is really interested in this connection between metabolism and cell fate decisions, both in normal cells and in cancer cells. And so just to introduce this concept, what does metabolism have to do with cell fate? How are these two processes even related at all? Well, I think in many ways, perhaps it's intuitive. If you think about the different cells in our body, liver, muscle, they all have different metabolic profiles. They have different jobs to do. Those different jobs to do require different metabolic profiles. But what's really important about this is that those metabolic programs aren't spontaneously acquired on terminal differentiation. They actually can be developmentally specified. Because you can see this even in a dish. Different cell types have different metabolic programs, even when they're stripped of the architecture and microenvironmental factors around them. And you can see if you take a stem cell and you have it commit to a lineage and differentiate, even in a dish, its metabolism changes. And so this shift in metabolism during developmental trajectories raises two questions that are the central focus of our lab. The first of which is, can metabolites actually affect these lineage decisions? Can they affect the likelihood of a cell committing to one lineage or maintaining a self-renewal program? So how could this be? How could metabolites contribute to cell fate regulation? There are many hypotheses for how this might be, but one of the ones that we think about a lot in the lab is the notion that metabolites can actually influence gene expression programs. That's because, of course, ultimately cell fate is specified by the complement of genes that are expressed in the cell. That determines who a cell is. And gene expression programs, of course, are ultimately controlled by transcription factors, but the transcription factors, the likelihood that these transcription factors can act and execute a gene expression program is in turn shaped by the chromatin landscape, the chemical modifications on DNA and histones that determine whether or not a transcription factor can access its cognate targets and turn on a gene expression program. And I think there's been a lot of interest, a lot of amazing work done, of course, in the Dana Farber area about how these chemical modifications can have critical implications for gene expression programs and cell fate specification. The reason we're interested in these modifications is because they are metabolites, full stop. These modifications are metabolites. That, for example, methylation is a methyl group that is deposited through the action of methyltransferases using a metabolite acetylmethionine as an obligate methyl donor. Analogously, histone acetylation or any acylation is donated by acyl or acetyl-CoA that's conjugated onto histone lysine residues through histone acetyltransferases. So these modifications are taking metabolites from intracellular metabolism and putting them onto modification to modify DNA and histones. The removal of these modifications also can depend on cell metabolism. The best example here is a family of proteins known as alpha-ketoglutarate-dependent dioxygenases. These enzymes use alpha-ketoglutarate and molecular oxygen as co-substrates to affect a hydroxylation reaction that can be an intermediate in DNA or chromatin demethylation. That results in succinate and carbon dioxide as byproducts. These metabolites come from central metabolic networks. So acetylmethionine, for example, comes from serine or methionine metabolism, really essential amino acids for many of our cellular processes. And alpha-ketoglutarate, succinate, and acetyl-CoA are all intermediates of the TCA cycle or the Krebs cycle, which sits in the mitochondria and is our terminal hub for nutrient oxidation. The TCA cycle is this really critical link between the nutrients that we have and that we burn to get energy, and also for making the precursors, the backbones of metabolites that we need to grow. So in this way, the TCA cycle really integrates what we have and what we need. And the idea then that these intermediates of the TCA cycle might exit the mitochondria, communicate to the nucleus to affect gene expression program, raises the notion that the nucleus might actually be in communication with the mitochondria to understand the metabolic resources of the cell at any given point in time and to attune gene expression programs accordingly. It's a cool hypothesis. It's still really a hypothesis, and a lot of labs are doing a lot of work on some of the biochemical and physiological challenges inherent in the hypothesis. But one thing I want to talk about is just the logic of this. If for this to work, for metabolites, these metabolites to be able to control cell fate programs in a reproducible way that's meaningful for development, there would have to be predictable changes in the steady state levels of these metabolites that are associated with cell state. Specific cell states would have to be associated with specific levels of these metabolites. Otherwise, you would have no ability to affect and maintain a coherent program of gene expression. And so then if these metabolites have to be sort of coherently regulated in a cell state specific manner, that requires that the metabolic networks that determine levels of these metabolites also have to be regulated in a cell state dependent manner. Otherwise, you're just going to have changes in these metabolite levels inducing gene expression programs on and off, and you wouldn't have coherent cell fate changes. So the best example that these metabolites can help exert coherent cell fate changes actually comes from cancer, which in many ways, of course, is a disease of aberrant cell fate regulation. A common paradigm across many human cancers is the notion that a cell that should have differentiated or has the capacity to differentiate doesn't differentiate the way it's supposed to and gets locked in sort of a progenitor-like state. Often this progenitor state can be maintained by hypermethylation of genes that would normally turn on terminal differentiation programs. So you're sort of locked in an aberrantly self-renewing program. And so that's led to some interesting ideas that perhaps these alpha-ketoglutarate dependent dioxygenases that can affect removal of methylation might control the fate of cancer cells. This idea has actually really powerful genetic evidence. So the product of these alpha-ketoglutarate dependent dioxygenases, succinate, is actually a competitive inhibitor of these enzymes. It competes for the active site with alpha-ketoglutarate and blocks their activity. There are a family of familial cancer syndromes, which are driven by germline mutations in the genes that encode subunits of the succinate dehydrogenase complex. This is the complex that consumes succinate in the mitochondria. These cancer predisposition syndromes are associated with cancers such as pheochromocytoma and paraganglioma. And what's really important about these cancer syndromes is that the tumors that arise in these patients are marked by loss of heterozygosity of the remaining wild type allele, which induces complete loss of function of the succinate dehydrogenase complex and extremely high accumulation of this metabolite succinate. This high accumulation of succinate is associated with inhibition of these alpha-ketoglutarate dependent dioxygenases in these patients. They have high levels of HIF, which is also targeted by these enzymes. They often have hypermethylation as well. And so this really mechanistically links these pathological alterations in a metabolite with the aberrant cell fate change that is predisposing these patients to have tumors. It's not just succinate. Fumarate is a closely related metabolite to succinate that can also serve as a competitive inhibitor of these enzymes. And it also is associated with familial cancer syndromes, again, where there's one germline mutation and the tumors that arise in the patients are marked by loss of heterozygosity of the remaining allele. I think what really sort of broke open this case and made it quite clear that pathological alterations in metabolites could induce a persistent steady state or a persistent fate change that was predisposing cells to tumorigenesis was the advent of whole genome sequencing that revealed very, very common mutations in isocitrate dehydrogenase 1 and 2. These mutations are not loss of function, they're actually gain of function. They're neomorphic and they allow isocitrate dehydrogenase, now these mutant forms, to reduce alpha-ketoglutarate to a structurally similar metabolite 2-hydroxyglutarate that looks a lot like alpha-ketoglutarate and can come in and bind in these active sites, but can't be used as a substrate. So it's also a competitive inhibitor. And altogether, what's become clear from these patient data is that high levels of these metabolites can induce hypermethylation, can induce an aberrant stem-like phenotype, and can predispose to tumorigenesis, arguing that pathological accumulation of metabolites really can alter cell fate and push cells towards a more self-renewing phenotype. When we started the lab, we were interested in this other side of the equation, which wasn't talked about a lot, which is this alpha-ketoglutarate itself. Since these are all competitive inhibitors with alpha-ketoglutarate, we were curious as to whether levels of alpha-ketoglutarate itself had any say in the balance between self-renewal and differentiation. Could the availability of alpha-ketoglutarate affect the likelihood of a cell to engage in differentiation that might in some ways be tumor suppressive, perhaps in some lineages? We actually got into this question somewhat serendipitously. We were interested in alpha-ketoglutarate and we were also interested in p53. And then the projects sort of magically came together in work that we did in collaboration with Scott Lowe's lab, led by John Morris, who now has his own lab at University of North Carolina, and Josie Yashinsky, an amazing grad student in my lab. We used some beautiful models of p53 and in mouse pancreatic ductal adenocarcinoma. I'm not going to talk about the models because this work was published now several years ago, but essentially we used a cool model that Scott's lab has made where tumors are driven by inducible hairpins against p53, which allows you to create a tumor, in this case a PDAC tumor, that doesn't have functional p53 unless you withdraw DOX and you silence the hairpin, and p53 will turn on and the tumor will mount an intact p53 response. And we loved this model because we would get tumor suppression. You could control reversibly when you want to have a cell turn on a tumor suppressive response, which is a fate change. And we love fate changes we can model. And we asked, what is the metabolic change that happens when you mount a tumor suppressive response? And the big change that we saw was alpha-ketoglutarate was massively going up. So we found that p53 directly can control accumulation of alpha-ketoglutarate in these cells. Moreover, alpha-ketoglutarate was sufficient to induce differentiation programs, even in cells that didn't have p53. And we could override this p53 response just by putting in high levels of succinate. Altogether, which told us that p53 can control differentiation, can control tumor suppression, in part by activating this axis, pushing the cells the other way. And that told us, moreover, that a really common feature of all these tumor suppressors, p53, fumarate hydratase, succinate dehydrogenase, are all bona fide tumor suppressors. A common feature is creating a metabolic landscape that's permissive for self-renewal. It changes levels of these metabolites that tips the balance of this reaction towards a hypermethylated self-renewing phenotype. And we think this is what IDH mutations are doing. They're co-opting this endogenous tumor suppressive axis to lock cells into a hypermethylated state. But what I want to talk about for a few minutes is that we don't think it's just genetics that determine whether or not you can activate this axis. This axis does seem to be responsive to some environmental cues. And in particular, in work that we did in collaboration with Elaine Fuchs's lab, led by Sanji Bash, who's now a resident at MGH, and Pavlina Todorova, a great postdoc in the lab, we found that circulating nutrient levels can also control the likelihood that this reaction can occur. So we came into this by studying epidermal stem cells. Epidermal stem cells are relevant because they are the cell of origin for squamous cell carcinoma. They are also, of course, the homeostatic stem cells that maintain our skin. And we love them, again, as a model system because we can culture them in a dish and we can change their fate to make them oncogenic or not oncogenic, and we can ask what happens to their metabolism. When we looked at the metabolism of epidermal stem cells, the number one feature that jumped out to us was that these cells really like to take up exogenous serine. They were very good at taking up exogenous serine and using that to fill their intracellular serine pools. We found that then if you removed serine, actually serine is a non-essential amino acid, and if you remove it, cells are forced to make it on their own through a pathway from taking a glycolytic intermediate and converting it to serine. Notably, this pathway also makes alpha-ketoglutarate as a byproduct. And what we found is that when we withdrew serine from epidermal stem cells, they differentiated. And so I'll show you what that looks like here, where we could take epidermal stem cells in a dish, stain them for keratin 14, which is a marker of a stem-like state. And we found that if we starved them of serine and the closely related amino acid glycine, they differentiated. They lost expression of this stem cell marker. And importantly, this was not because of serine itself. Serine's a methyl donor. This has nothing to do with serine itself. It's actually the activation of the serine synthesis pathway, which we could see by silencing the first enzyme in this pathway, and then seeing that these cells no longer differentiated. So it wasn't serine itself, it was activation of de novo serine synthesis that was inducing differentiation. And that was because de novo serine synthesis forces cells to accumulate alpha-ketoglutarate. And you can see here that just by adding cell-permeable alpha-ketoglutarate, we can restore differentiation downstream of PHGDH silencing, consistent with rescuing a downstream step in a pathway. And just to show you what this looks like in vivo too, this is actually really relevant to our homeostatic epidermal stem cells. We can starve mice of serine and glycine. This is a diet pioneered by Karen Boutzen. It's shocking that it works, but it works very well. You put these mice on the serine glycine deficient diet for a few weeks. What we found is that the skin actually thickened. You can see here shown to keratin 10, a marker of the differentiated cells. They're becoming much thicker, and we can use a reporter of differentiation to show that this is driven by precocious differentiation of the epidermal stem cells. So exogenous serine availability was a critical determinant of stem cell fate, and this had really important implications for cancer. So Karen Boutzen, again, was the first one to show that if you starve mice of serine and glycine, xenograft tumors don't like to grow as much, and we can see this as well with allografts. Normally, these are squamous cell carcinoma allografts. They grow very, very well in the dermis of these mice, but if you put the mice on the serine glycine-free diet, the tumors don't grow at all. Actually a really, really shocking dietary effect right here. And I think this has to do with the fact that the skin loves to eat serine. It gobbles it up, it really needs it. And so it has a very dramatic effect when you remove serine from the diet. What you can see is that when we put these mice on a diet, it's the stem cells in the tumor that are preferentially depleted. Here we're staining stem cells with CD34. You can see control tumors are sort of replete with stem-like cells. And if you put the mice on the diet, now it's these stem-like cells that are really getting outcompeted, and that's because they're differentiating. So our model is that serine availability controls epidermal stem cell self-renewal by determining whether or not cells accumulate alpha-ketoglutarate downstream of the serine synthesis pathway. And so to test if this is what's really why the diet is tumor suppressive in this lineage, we took mice, put them again on a controller serine glycine-free diet, and then we took squamous cell carcinoma cells where we'd genetically enforced alpha-ketoglutarate or succinate levels as a way to test tipping that balance to see if it's, again, this dioxygenase activity that's responsive to dietary levels of circulating amino acids. And then we could see what happens to the tumors that form. So again, this is published. I'm just going to show you this one key experiment, which is what happened to tumors on a serine glycine-free diet. Control tumors growing in mice on a control diet were chock full of undifferentiated cells, shown here staining for keratin 14 in gray, and they had really not many differentiated cells at all, which is shown in keratin 10 in red. But when these tumors are now grown in mice starved of serine glycine, the tumors differentiate. You can see just this really, really clear switch where they now are staining much, much higher for these differentiated keratin 10 marker. If we genetically enforce alpha-ketoglutarate accumulation by essentially messing with its consumption in the TCA cycle, the tumors differentiate even on a controlled diet. So alpha-ketoglutarate is sufficient to induce tumor suppressive differentiation, not only in pancreatic cancer, but also in squamous cell carcinoma. And this is the experiment that sort of blew my mind, didn't think it would work, is that if we did the inverse and we genetically enforce succinate accumulation by targeting succinate dehydrogenase complex, now the tumors remain undifferentiated even on the serine glycine-free diet, and they continue to grow just fine. The serine glycine deficient diet is no longer tumor suppressive in this case. And I think this sort of raises a few important points, one of which is different lineages are going to have different metabolic dependencies. Dietary levels of nutrients can have a really important effect on tumor growth. But third, the effect of these dietary nutrients on tumor growth is going to be dependent upon the genotype of the tumor itself. Whether or not the tumors will respond to diets is going to depend on why that diet might be tumor suppressive or tumor promoting, and whether or not the tumor has an intact axis to respond to the diet. And in this case, in this lineage, this diet is tumor suppressive because it induces differentiation. But if you have a genetic mutation that prevents you from responding to that differentiation cue, the diet is no longer tumor suppressive. So overall, this is sort of why we're interested in this notion that metabolites can actually control cell fate. They are continually surprising us for their ability to really dictate whether or not a cell can engage in a productive cell fate transition. But it's gotten us also interested in a somewhat broader point, which is for any of this to work, different cells have to have different metabolic profiles. And you can see this in the skin really clearly. These skin cells really, really, really like to take up serine. They don't like to make it. Being able to take it up helps preserve them in a stem-like state, whereas their differentiated counterparts now are very good at making serine. So there are very clear, specific cell state dependent metabolic programs. And we've become very interested in trying to understand what those are. How are cell type specific metabolic programs established? And if we understand them, can we really exploit them and go in there and enrich for cells in desired stages of development, essentially tip the balance between differentiation or self-renewal at will by understanding the metabolic demands of cells in one or the other state. So overall, if we take a step back and think about what is metabolism for, well, metabolism is for many things, including cell growth. That's often what we look at when we look at cancer cell growth or cell growth in a dish. We're looking at the ability of a cell to generate biomass, which requires making nucleotides, lipids, ATP. And any cell of any lineage that's growing is going to need these things to a certain degree. But there are differences. And for example, as I just told you about in the skin, even though every cell is going to need serine if it's going to grow, how it gets that serine is different. Sometimes cells prefer to take it up. Other times they prefer to make it. And what determines that difference? What determines whether or not you want one or the other, I think is really not very well understood. And so we've started to think about metabolism less as sort of a cookbook recipe that every cell needs to follow this set of reactions to get a productive growing cell, and more of a hodgepodge of options. And so if you think about metabolism as a set of Legos, maybe there are different ways to grow. You could take all the blue Legos and you can make one tower, and that's one way to grow. Or you could take all the red Legos and you can make another tower, and that tower might look a little different, but it's still a tower and you're still growing. So the idea here is that there might be different ways to assemble metabolic networks to meet the demands of the cells. And what we wanted to know is, what are those different ways to assemble metabolism? How can metabolic networks come together to help cells make their specific cell type, essentially tower of Legos? And we realized that a really good tool to begin to ask this question was DepMap, the cancer dependency map made out of the Broad Institute. This resource, I'm sure most of you know about, has sequencing, profiling, and importantly, CRISPR screens for almost 1,000 cell lines now at this point. And the reason that CRISPR screens are so useful for us for thinking about how cells grow is that by definition, these CRISPR screens are, the readout is growth. The readout is how much does any given gene support the growth of any given cell line. So 2 students in my lab tackled this question, trying to understand, are there specific programs that different cells use to grow? And this was led by Paige Arnold, an amazing graduate student who's now off at her postdoc at Rockefeller, and Ben Jackson, an amazing MD PhD student who's finishing up still in the lab. And they looked at metabolic genes in DepMap. They looked at how much different metabolic pathways are required for growth. What you can see here is we took 4 central metabolic pathways, which we've colored in different colors. And every dot here is now an average of all 800 or so different cell lines. Up here is the expression, how much any given gene is expressed, and here's essentiality. The further out you are in the x-axis, the more essential you are to grow. The more a cell cannot grow if you do not have this gene. What you see is there's a real big heterogeneity. Some genes are very poorly expressed overall on average, but even among the genes that are expressed, there's a big range in how much a cell needs a gene to grow. And that's true even for pathways. If you take glycolysis in yellow here, some glycolytic enzymes are essentially dispensable, whereas some are very important. So that tells you that there's no one pathway that's required for growth, full stop. That's not true, but rather that there are parts of pathways that are more or less essential. But the real power of the system comes when we don't just take the average level. I would say anything around here off would be considered essential and would just count as, these would be essential genes. That's important, but it's lost a lot of the power of the system. The power of the system comes from looking at the heterogeneity itself, looking at how much a gene is necessary across each of the individual 800 cell lines. As an example here now, I'm plotting just the essentiality data of the TCA cycle genes, because the TCA cycle is this essential hub of nutrient oxidation and biosynthetic growth. And what you can see here, these violin plots are showing all 800 lines, that there's a range in the essentiality. These genes over here are essential, these genes are non-essential, but even within these broad categories, there's nuance. So for example, IDH2 is a neutral gene, it's not essential. And by and large, most cells don't care whether or not you have IDH2. If you go over here though, to the essential genes, there are a lot of cells that really, really care whether or not you have these essential genes. But even so, there are cells that don't care at all. So if you look over here for SDHD, there are some cells that even might grow faster if you don't have SDHD, even though it's considered an essential gene. And what that tells us is that, again, there's more than one way to grow. different cells have different strategies. They have different nutrient needs, they have different pathway needs. And so what we wanted to do was to try to understand, what are these different strategies? How can different cells grow in different ways? So there's a lot of power in this heterogeneity, which we can exploit. And as an example, I'm going to show you SGHC and SDHD, which were those two most essential TCA cycle genes. If you plot each cell line, the essentiality of these genes, you see that they're highly correlated, which intuitively makes sense. SGHC and SDHD are two membrane-bound subunits of the succinate dehydrogenase complex. They physically work together. And so if you need SGHC, you're also going to need SDHD, and vice versa, because they essentially are one functional unit. And so you can plot all 800 lines and get a Pearson correlation score that's quite high. And what many people have realized, first David Sabatini's lab and now many others, Michael Bassick and others, were doing really beautiful work here, showing that these codependencies, genes that have functionally correlated dependencies, are genes that work together in functional modules. It's a really, really powerful system. And if anyone who hasn't used it before, I highly recommend you try it. It's a lot of fun to play with your genes. And if you didn't know anything, you could go in, for example, and sort of predict the whole mTOR network, or at least many key parts of the mTOR network, by looking at genes that have functional correlated dependencies. So we wanted to do this with metabolism. We wanted to know what parts of metabolism work together. And in that way, we figured we could start to understand why different cells grow in different ways. So we took our genes of interest, we did these pairwise correlations, and then we made a correlation matrix to look for nodes of groups of genes that work together. This is what it looks like if you do all of these genes that I just showed you across these 4 pathways, and you see there's a lot of white space. That's probably because these genes either are poorly expressed, or there's a lot of isoform redundancy, because this is the one weakness of CRISPR screens, is it really won't work if there are functional redundancies. But clusters do emerge. You can see there's one cluster here of glycolysis. There's one cluster here of 1-carbon metabolism. There's one cluster here of the TCA cycle. But then there's this other cluster here that was also the TCA cycle. And that really confused us, because the TCA cycle is this, one pathway that we draw in textbooks all the time. So we wanted to look at these data a different way. And so we plotted it here, exact same data, but now plotted in 2 dimensions, where the distance between any 2 genes and the thickness of the line connecting them is a representation of how tightly correlated they are. You can see here's our glycolysis cluster, here's our 1-carbon metabolism cluster, and here in red is our TCA cycle cluster that's coming apart in 2. It's separating into 2 separate functional clusters, linked at the middle here by this gene DLD. And this so satisfying for me, because DLD is an obligate subunit of both the OGDH complex, which sits right here, OGDH and DLST, and the PDH complex, which sits right here. And so this is, again, I think just the power of DepMap for being able to show us how things work together. You can see that DLD has these split loyalties because it's a part of both of these complexes that otherwise reside in separate functional units. So we've called these, we decided these were sort of 2 separate clusters, and we wanted to see what they were. Why were these separating in 2? Mapping this cluster on the traditional TCA cycle gave us a really surprisingly clean answer. Every gene here is now highlighted in red, in bold red. And it became clear that all of the genes in this cluster were the genes required for oxidation of citrate in the mitochondria. This is the TCA cycle for those who don't obsess about it as much as we do. So everything required for the burning of citrate in the mitochondria was in this cluster. If we then looked at this cluster, it was everything required for making citrate. So these functional clusters were splitting the TCA cycle really neatly down into 2 separate halves, one required for making citrate and one required for burning citrate, which raised 2 questions for us. First, why wasn't citrate production tightly functionally coupled with citrate oxidation? Was there something else that citrate could be doing? And 2, who was making citrate? If citrate production wasn't tightly coupled to the regeneration of the carbon backbone needed to continue to spin the cycle, who was regenerating the backbone? How were you making citrate in the first place? The answer to this question came when we looked at this other gene that came along for the ride with the TCA cycle cluster, SLC25A1, which is a mitochondrial transporter that exports 1 citrate to the cytosol in exchange for 1 malate. That made us realize that instead of oxidizing citrate, this transporter could take citrate out into the cytosol, where it could be cleaved by ACL, or ATP citrate lyase, forming oxaloacetate that then is 1 easy reduction step away to malate, which neatly completes the cycle. So So we're not the first to think of this. Piet Borst actually made this up, I think, when he was a grad student, which is humbling to the rest of us. He called it the citrate malate shuttle. And we realized, though, that it proposes a way to get reducing equivalents into the mitochondria, because this reaction here will pick up an electron in the cytosol. But we thought that maybe this isn't really just an electron shuttle. The fact that it's coupling so tightly with citrate production across 800 cancer cell lines made us realize that maybe this is actually an alternative fate of citrate, and maybe an alternative way to regenerate this carbon backbone that's required to sustain the TCA cycle. So is this true? Is this alternative fate of citrate, this alternative TCA cycle, a major route of metabolism in cells and culture? Now, there are a couple ways we can look at this. One way we can do this is to give cells uniformly labeled glucose, which will go through glycolysis and ultimately make pyruvate, which can be important to the mitochondria. And when it kicks off the TCA cycle, this will result in a citrate that has 2 heavy labeled carbons. If this citrate goes through the canonical TCA cycle, the carbons that are burnt on this first turn are not the labeled carbons. It burns the unlabeled carbon. So any malate or downstream metabolite that's made from citrate in this canonical mitochondrial TCA cycle will also have 2 labeled carbons. But if citrate goes out into the cytosol and is cleaved by ACL, ACL liberates those labeled carbons, and any metabolite that's made downstream will be unlabeled. So the first thing we wanted to see is there a drop-off in labeling? Do we lose label between citrate and malate when we look in cancer cells? We took advantage of a really beautiful paper from Ralph DeBardinis' lab that came out several years ago, where they profiled over 80 non-small cell lung cancer lines. And when we looked at this N+2 labeling of metabolites, we see that citrate is highly labeled. And you see, again, there's a range. There's metabolic heterogeneity, which is what we're interested in here. But that label is lost. It goes down as you start to make these downstream metabolites from fumarate and malate, suggesting that indeed there might be this other route of citrate metabolism. So to represent this drop-off, the degree to which malate is made from this citrate, we use this ratio of malate N+2 to citrate N+2. It tells you the fraction of malate that came from this M2 citrate in this canonical TCA cycle. This ratio is quite low. It ranges from about 0.05 to 0.5. And what we wanted to know is this label loss mediated in part because of ACL, which is pretty easy to test. We can add an ACL inhibitor to cells and then see what happens to this labeling. And what we found is that, that label now really goes up. The fraction of malate that is made from citrate in the mitochondria goes way up if we inhibit ACL, which is consistent with the notion that ACL is a driver of this label loss downstream of citrate. For any aficionados who might be on the Zoom, there's another explanation. You're probably chomping at the bit. There's a trivial explanation for this label loss downstream of citrate, which is that in this experiment here, glutamine is coming into the TCA cycle as a major anaplerotic source, and it is unlabeled. So if malate is coming from glutamine, it will be unlabeled. The reason I really like this experiment is that the ACL inhibitor didn't reproducibly affect glutamine anaplerosis across these cell lines. And so while glutamine anaplerosis is absolutely a major reason for this label dilution, and it's probably why this ratio rarely gets above 0.5, this effect of ACL is independent of that and argues that ACL really is mediating a significant fraction of this label loss. So it's not just cancer cells. We can also do this in mouse embryonic stem cells, which we like to use a lot in the lab as a model of cell fate transitions. And again, you see that citrate can be very highly labeled from glucose. Malate is much less labeled. And that drop off, again, is partially mediated by ACL. We made genetic clones to test this, the specificity. And now I'm showing you all of the metabolites and their labeling patterns. Again, you see ACL loss doesn't really affect the ability of glucose to make citrate in the first place, but it does affect what happens to citrate. That citrate is much more likely to be able to process to make malate in the TCA cycle if ACL is not around, which you can see is this increase in the ratio of N+2 malate to citrate 2. So this raises the possibility that the TCA cycle can transit through the cytosol, and we wanted to be able to see if we could test that directly. There are tools to do this. We were inspired by work from Matt Vanderheiden and Christian Metallo, where you can use a form of glucose where instead of the carbons being labeled, there's a deuterium on the 4th carbon. You have a hydride label instead. When this glucose goes through glycolysis, up to 1/2 of the triose molecules will have this heavy label. And when that is now acted upon by GAP-DH, the reduction step here will transfer that hydride onto NADH. What that allows you to do essentially is to track redox reactions in the cytosol. Anybody who picks up this electron from NADH will actually pick up the hydride label. You can see that because lactate, which is reduced, the reduced form of pyruvate, is labeled at up to 30% in the cells. In theory, it should be up to 50% because it's half of glucose, but in practice, it's 30%. So we reasoned if this is a way to track NADH-dependent reactions in the cytosol, we could see if indeed malate is being made in the cytosol using this tracer. And that's exactly what we see. Our results match very well with, again, work from Matt and Christian, where you see that malate is actually as labeled as lactate, arguing that a significant portion of lactate is generated from a carbon backbone in the cytosol. And with most important thing, though, to see if this is actually a TCA cycle is to see if that malate goes back into the mitochondria and is used to make citrate. If malate is reimported, it equilibrates with fumarate. And if that fumarate goes forward and is used to make citrate, that citrate would also have the hydride label. Oops, sorry, my animation was off. And it does. There's the citrate, it also very labeled from the hydride. So our next question was, if malate can go back into the mitochondria, is any of this dependent upon ACL? Is ACL actually kicking off this cytosolic TCA cycle? And indeed, you see that if we knock out ACL, the fraction of malate that is made in the cytosol is reduced. There's less malate made in the cytosol, arguing that ACL is a source of cytosolic acetyl-CoA. And importantly, the amount of citrate that's regenerated from this label is also reduced. So the TCA cycle can transit through the cytosol in an ACL-dependent manner. And then we wanted to know if this was actually important for TCA cycle dynamics. So we just looked at steady state levels of the TCA cycle metabolites in our embryonic stem cells. And really, to our surprise, the answer was very, very clear, which is that when we didn't have ACL around, the upstream substrate citrate increased massively, and the downstream metabolites malate, fumarate, and aspartate all decreased massively. This is really consistent with strangling a pathway right at the step of ACL. You see the upstream up, the downstream down. And gratifyingly, alpha-ketoglutarate and succinate, which are canonical TCA cycle metabolites, were unchanged, really showing specificity of this perturbation. These results are really big. This is a log fold change. We were really shocked by this result, and we were especially more shocked because we did the control and we knocked out aconitase 2, which is the enzyme in the canonical TCA cycle. And when we looked at steady state metabolite levels, nothing really changed at all in cells. Cells essentially was metabolically inert in our cells to knock out ACO2. Which gave us a lot of pause because this is not the TCA cycle that Krebs described. And actually at this point, lockdown happened and we had a lot of time to think about our data and a lot of time to think about what it might mean. So Paige went and reread all the original Krebs papers to really understand what did Krebs show and how did he show it, to make sure we hadn't missed something and we weren't doing something stupid. So Paige was really inspired by reading Krebs's work because he talks a lot about how his big breakthrough for identifying the TCA cycle came when he turned to using pigeon breast muscle as a model system. It's an extremely oxidative tissue, and so it was a very fruitful place for a lot of these researchers to elucidate these critical oxidation reactions. So we thought, well, if muscle's where it's at, let's look at muscle. And so we took myoblasts, a progenitor cell, and we could differentiate them into myotubes in a dish. This works beautifully, as you can see here. And then we looked at metabolism in the myoblast state or in the myotube state. And what we saw was a very dramatic switch, whereas in the myoblast state, cells had a very low malate +2 to citrate +2 ratio, consistent with a non-canonical TCA cycle, but that ratio went very high when they differentiated, consistent with a switch to canonical. And so to see if there really are these two different cycles that are engaged in a cell state dependent manner, we used DOX-inducible hairpins to knock down either ACL or ACO2 in the myoblast or myotube state. What we found is that ACL seemed much more important in the myoblast state. When we knocked down ACL, there was a bigger effect on the malate 2 to citrate 2 ratio and a bigger effect on steady state TCA cycle metabolites when cells were in the proliferating myoblast state. That effect on steady state TCA cycle metabolites and this ratio really was diminished dramatically when cells became myotubes, arguing that the reason we'd seen such a big effect of ACL on the TCA cycle was because of the state of the cells, and that if we looked in muscle like Krebs, it really was going away and it wasn't there as much anymore. So this led us then to ask if there can be really fundamental differences in metabolic networks depending on their cell state. Is this required? Do you have to pick the right TCA cycle to be the cell that you want to be? essentially. So the model system that we really like to use to ask these sulfate transitions are embryonic stem cells, because it's their job to undergo these sulfate transitions. They're extremely good at it. And thanks to really amazing work from pioneers in the field like Austin Smith and others, they're extremely well worked out genetic and media formulations to induce cells into one state or another. Up till now, everything I've shown you in embryonic stem cells has been in a traditional medium formulation containing serum and leukemia inhibitory factor. This maintains cells in a pluripotent state, but they are metastable, they're heterogeneous, they're prone to spontaneous differentiation. And epigenetically, they look more actually like a post-implantation-like state, not a pre-implantation-like state. Austin Smith's lab discovered that you can add 2I, these two kinase inhibitors, an inhibitor against GSK3 beta and MEK, on top of this media formulation. And that drives cells into a very homogeneous, highly self-renewing population that looks a lot like the pre-implantation epiblast. It's hypermethylated and everything looks good. So this is what's thought to represent sort of the naive or ground state of pluripotency. And then this becomes a really nice tractable model system where you can withdraw 2I lift, the factors that are sustaining pluripotency, and the cells will dismantle the pluripotency network and acquire differentiation competence. And I'll show you what that looks like. It's a very reproducible stepwise manner. As you withdraw lift over time, cells downregulate pluripotency genes, and they upregulate genes that are markers of a more post-implantation or differentiation competent state. That means then we can assess their functional capacity. If you try to return them at the end of this assay back into 2I media and ask if they still can maintain self-renewal capacity, they retain self-renewal capacity for the first 12 or so hours, but after that, they really lose it. And so they've exited the pluripotent state. So we wanted to know what happens to metabolism in these different states. The first thing we did was look in the naive state and induce it to engage differentiation competence with our glucose tracer. We found that this malate plus 2 to citrate plus 2 ratio was extremely high in the naive state. They were as high as myotubes at being extremely canonical. And they went from canonical to non-canonical as they started to differentiate or gain differentiation competence. And we see the reciprocal with our cytosolic glucose tracer that traces non-canonical TCA cycle activity. We see the opposite. We see that, again, they have very little TCA cycle transiting through the cytoplasm when they're in the naive state. And as they gain differentiation competence, now the TCA cycle is going like crazy through the cytoplasm. So there's this switch from canonical to non-canonical TCA cycle metabolism as you exit pluripotency. And so we've asked what happens. We reasoned that if they're truly switching cycles, you should have different genetic dependencies, which is sort of how this whole project started. So if we took our ACL knockout cells, the same cells I've shown you previously in serum LIF, and we converted them into the naive ground state, it looked pretty much genetically inert. There was no effect on the malate 2 to citrate 2 ratio, no effect on steady state TCA cycle metabolites anymore, because these are cells doing the canonical TCA cycle. They don't care about ACL. And that means if you look at anything, their ability to make protein, to proliferate, to grow, ACL doesn't matter. They do not care about ACL in this naive ground state. But now, if you ask them to exit pluripotency, naive pluripotency, and become differentiation competent, that requires engaging the non-canonical TCA cycle. And so ACL becomes required. You see that now, dismantling naive pluripotency unmasks the effect of ACL on metabolism. You have a big effect on the malate 2 to citrate 2 ratio, a big effect on steady state TCA cycle metabolites. And that means that this metabolic reorganization that's required for cells to meet their new cell state, or that accompanies cells as they meet their new cell state, can't happen. They can't induce their malate, they can't induce their aspartate if they don't have ACL. So altogether, this is, I think, a nice example of how metabolic rewiring accompanies cell state transitions. We can identify genes that mediate that metabolic rewiring. We can knock it out, and then that can reveal whether or not that metabolic rewiring is required, which in this case, it is. If you cannot engage the non-canonical TCA cycle, you cannot exit the naive pluripotent state. You can't sustain protein synthesis, you can't sustain proliferation, and you die, which we can also show another way using a pharmacologic inhibitor of ACL. We can use a reporter of the naive pluripotent state where a GFP is knocked into the endogenous REX1 locus. This is made by Austin Smith and is a kind gift from him. It's a really beautiful reporter of the naive pluripotent state. Here in gray is when cells are cultured in 2I. That reporter is high and uniform. But when you withdraw 2I LIF after 40 hours, almost all the cells are in this REX1 low state. So you're monitoring whether or not a cell is in or out of the naive pluripotency. And to our surprise, if we block metabolic remodeling with an ACL inhibitor, it was as good as the gold standard 2I-LIF at maintaining cells in a REX1 high population, arguing that if you don't have ACL, you just can't do it. You can't exit the pluripotent state, or if you do exit, you die. We're not sure which it is. But either way, you can't do it. You can't exit the pluripotent state. And we can also see this genetically. If we knock out ACL and ask cells to exit pluripotency, they can't do it. So they maintain high levels of these pluripotency transcription factors. They can't induce differentiation-associated genes. And that means they actually retain the capacity for self-renewal. If you reseed these cells and ask, are you still self-renewing? The cells that couldn't activate the non-canonical TCA cycle still can maintain self-renewal, but those that activated the non-canonical TCA cycle can't. And so there's one explanation perhaps for this change in cell fate, which is this idea I introduced earlier that metabolites can control the chromatin landscape. And in particular, I'm sure some people have spotted that ATP citrate lyase makes acetyl-CoA in the cytoplasm. Katie Wellen and Craig Thompson were the first to show that this can provide a major source of acetyl-CoA for histone acetylation. So one hypothesis is that by knocking out ACL, you're compromising histone acetylation, which is what we see. And that's why you can't exit pluripotency. And it has nothing to do with metabolic networks, but really it's all just about chromatin. Luckily for us, there's an easy way to test this. You can add sodium acetate to your cells. They take it up like crazy. They can rescue the histone acetylation. You can rescue the lipid synthesis. Everything downstream of acetyl-CoA you can rescue, except the effects on cell fate. So here again is our REX1 reporter. It's very low when you exit pluripotency. It's very high if you can't activate the non-canonical TCA cycle. And adding ACL doesn't affect that. If anything, adding acetate makes you an even better stem cell, which has been shown before as well by several other labs. And again, you see this with gene expression. So the effects of ACL on cell fate in this case are completely independent of the acetyl-CoA, but rather are because of this ability to sustain a TCA cycle that's required to meet the bioenergetic demands of a new cell state. And as a final test of the model, this was actually a reviewer question, it's an amazing reviewer question. The reviewer said, okay, well, if the non-canonical is required to exit pluripotency, do you need to access the canonical TCA cycle to enter naive pluripotency? Is this indeed required for naive pluripotency? The question being, if we take our ACO2 knockout cells, which were essentially phenotype free in the serum lift state, would they now have a phenotype if we tried to push them into the naive state? And we tried this experiment a lot. We kept trying, and every time we tried to do this conversion to 2I, something would go wrong. The cells would be really unhappy. Then they'd come out at the other end and they'd be clearly weird. Some genomics event, it's bizarre. We kept trying over and over. And then we realized, oh, this is the phenotype. These cells can't enter the naive pluripotent state very well. And if they do, something has gone wrong with them. And so to try to capture that, we harvested RNA at all the different passages during this conversion to 2I, this conversion to the naive state, and looked at the induction of pluripotency genes, naive pluripotency genes as cells were undergoing this transition. We found that not only were cell numbers lower as we tried to do this transition, as fewer cells could do it, but also the induction of these naive pluripotency genes was much lower. So altogether, I think this tells us that even at the earliest stages of pluripotency, these pre-implantation states, there are distinct metabolic phenotypes associated with distinct stages of cell identity. And those metabolic programs are often essential to becoming who you are. Metabolism is not just a passenger or a preference of who you are, but can be an absolute requirement of entering a certain cell identity. And I'd argue that that means that when we think about cell identity, we can't just stop at gene expression, but we also have to think about metabolism, and that it's a really part and parcel of who you are as a cell. And if you can't do the metabolism of a liver cell, then maybe you're not a liver cell. And so just to end, why does it matter which TCA cycle you do? Why do cells care so much about whether they do one or the other TCA cycle? I think is a really important question, because in the end, they're both TCA cycles. They both are regenerating oxaloacetate for the continued production of citrate. So why does it matter which one you do? And there are a couple key differences. The first of which is that the canonical TCA cycle is combustive. It burns your carbon as a way to maximize harnessing reducing equivalents to make ATP. That's a great strategy if you're a muscle cell. But if you're a cancer cell proliferating in a dish, or maybe some other cell types, burning all your carbon, making ATP is not necessarily your main goal. And actually often retaining that precious reduced carbon could be extremely beneficial to a proliferating cell or a cell that has a lot of demand to make lipids or histone acetylation. And that's what this non-canonical cycle allows you to do. It allows you to continually preserve these reduced carbons in the form of acetyl-CoA, which is our perfect sort of biosynthetic building block. The other major advantage of this cycle is that the non-canonical cycle is very difficult to sustain because that every one pyruvate that you put in the mitochondria and burn costs you one oxidized NAD in the cytosol. Normally, cells in culture get that NAD back by reducing pyruvate to lactate. That's the Warburg effect that we hear so much about. But that doesn't solve your problem if you're burning that pyruvate in the mitochondria. If you're reducing it to lactate, you're not burning that pyruvate. So there's sort of a zero-sum game here that makes oxidized NAD a major constraint on cell growth. And Matt van der Heijden and we and others have shown that oxidized NAD is a constraint on growth in cancer cells. This non-canonical TCA cycle solves that problem. Every one pyruvate that you put in the mitochondria costs you one oxidized NAD that you get back here in the cytosol through the reduction of oxaloacetate. So it becomes cytosolic redox neutral in a way that I think is going to turn out to be extremely beneficial to cancer cells and probably also stem cells as well. So with that, I'd like to thank everybody who helped with the work. The skin stem cell work was done in collaboration with Lane Fuchs, led by Sanjeev Bash and Paulina Todorova, and the TCA cycle work was done by Paige Arnold and Ben Jackson with some help from our good friends Lucas Sullivan and Fred Hutch. Thank you all.
Thanks, Lydia. That was really terrific work. I'd like to encourage the audience to put your questions into the Q&A tab and I will read them. And maybe I'll ask the first question, which is how is the switch from the canonical to the non-canonical cycle regulated?
I don't know. It can be different things in different cell types for sure. And that's something we're working on. There seem to be a couple things that seem to be conserved, but it sometimes can be as simple as the level of certain proteins. You know, if you give a cell more aconitase, it'll do more canonical. But I think what's really regulating the switch across different tissue types is probably slightly more complex than that. And it seems to be very tightly linked to cell state and the oxidative capacity of different cell states. So something we're working on.
Cool. Question about two different questions. Whether this pathway functions in other cell types, and what happens if you remove both ACO and ACLY?
Oh, I like that latter experiment. That just came up in lab meeting last week, and we're doing it now because that will tell us the epistasis of the cell fate, I think, a lot. If ACO2 unravels the ACL, anyway, we're doing that experiment now. It's a great experiment. I'd imagine the cells hate it, is what I'd guess, but we have to test. As to whether or not this is happening in other cell types, yeah, a lot. And there's heterogeneity in the degree to which it happens. So we came at it and found it because of DETMAP, which is cancer cells. So in general, cancer cells probably are doing this a lot more in a dish. Tracing data, comparing cells growing in a dish to growing as tumors in vivo suggests that cancer cells actually switch, and that when they're in vivo, they might actually be doing the canonical TCA cycle as well. So, which again gets at this idea that the non-canonical is a really great way to grow like crazy, which is what cancer cells in a dish are doing, but that cancer cells in a tumor are not actually necessarily growing like crazy, and they might switch back. So that's again gets at what's regulating the switch. And it shows you that even within a specific cell type, the same cancer cell line can do different cycles sometimes, depending on how it's growing. But we're looking a lot at this, looking at which cancers tend to use one more or the other, if cancer cell state determines one more or the other. And I think that's sort of the big new thing that we're working on now in the lab.
And maybe perfectly related to that, are there associations of this canonical versus non-canonical TCA cycle dependence in DEPMAP that gives you insights into where it might function?
Yeah. So that's a really good question. There are, and so there are two tricks. One is, I don't know if anyone caught this, but that ACO2 is one of the most differentially essential TCA cycle genes. So it's huge spread. And you can use that alone to find out sort of what drives one or the other cycle. And that's been fruitful for us. But the other is Ralph DeBardinas has this beautiful data set of 80 non-small cell lung cancer lines. And so you can line that up as well and look at gene expression programs that associate with one or the other TCA cycle choice. And what's been amazingly gratifying for us, brand new data, is that the answers that ACO2 genetic dependency gives you is the same as the answers that the metabolic tracing gives you, which again really argues that what we're looking at with this malate 2 to citrate 2 reporter is the degree to which you're using ACO2. And it seems to be the best answer we can get is cell state. The more stem-like a stem cell, the more that seems to be the biggest determinant of its TCA cycle. So we're looking at cancer cell differentiation and TCA cycle.
And then 2 questions related about whether lipid metabolism switches in the same way when you flip into this non-canonical cycle.
Yeah, so that is almost certainly a very important part of this. If you need to make a lot of lipids yourself, you're going to pull out a lot of carbon through ACL, and then almost by default, you'll be doing a lot of non-canonical. And that's certainly true. One of the best ways to induce the non-canonical is to starve cells of lipids, but you don't have to. So you can have changes in the TCA cycle, even if you aren't changing your lipid metabolism. So there's definitely a relationship. It can be sufficient, but not necessary.
And maybe we'll just take one more. If you restore the non-canonical TCA cycle after inhibited, is the effect reversible?
Oh, I like that experiment. Have we done that? I would guess so. But I'm going to write that one down. It's a really good one. I think so. I don't see why it wouldn't be. But that's a good question.
All right. Well, thanks again, Lydia. That was really terrific. And thanks everybody for joining us. See you next week.